Okay, reminder that the quiz is due by midnight tonight, so that is available on Canvas. It's covering everything from the last exam, so when we started going over cell theory and all the organelles and that stuff, up until what we got through on Wednesday. So none of this material is going to be on there because you could have already taken it potentially. It opened at midnight. So basically up through this slide, through the tonicity, the hypertonic, hypotonic stuff in cells. Okay. Here, so just as a reminder, because this can get a little bit confusing. Oh, whoops, next slide, there we go. It can get a little bit confusing talking, all of this terminology, it all kind of sounds the same. But the idea behind tonicity is just how much solute is there. And remember, by solute, you can put salt in there instead of solute if you want. That kind of makes it a little bit easier for you to understand how much solute there is inside of a cell versus outside of a cell. That's going to determine where water is going. Is it going into the cell, or is it pouring out of the cell? Or is it kind of evenly going in and out? Is it balancing, basically? So I showed you this slide at the end of class on Wednesday. And what I definitely want you to know are the differences between what plant cells like versus what animal cells like. So animal cells, that's a red blood cell. They typically want to be in an isotonic environment, which means that the amount of solute outside the cell is the same as inside. Then you just have this nice even movement of water into and outside of the cell. You don't have explosion or shriveling, basically. Plant cells are a little different because they have this really strong cell wall around their plasma membrane. So it's a very sturdy structure that we as animals do not have around our cells. Our cells are a little bit squishier. We don't have a cell wall. Because plants have a cell wall, they can sort of resist this explosion, essentially. So the idea behind a plant cell basically wanting to be in a hypotonic environment. So it wants to have more solute inside the cell than there is outside. That means water is going to be coming in. So water is moving towards where there's more solute, towards the hypertonic area. So this would be inside the cell would be hypertonic, and if the environment around it is hypotonic. So it's always one or the other. So they're in relation to each other. If there's more water pouring into the cell, that basically allows the cell to be nice and what we call turgid, which is just, I guess, solid or stiff or whatever other adjective you want to put in there. So that's the main difference between plant and animal cells. We're going to do a little practice right now to make sure you guys kind of understand this moving of water in and out. You don't have to write anything down or turn anything in. We're just going to work through a problem that's really similar to something I'll definitely ask you on an exam. So if we're looking at, it's so weird. This is a beaker, but you can't see the beaker on this on the projector. Anyway, this is a cell sitting in some kind of solution in a beaker. And we want to figure out where the water is moving. So there's water in the cell, there's water outside the cell, and there's also these solutes. So the sucrose, glucose, and fructose are all the solutes. So we know that water is going to move towards the hypertonic, where there's more solute, right? So we need to figure out, is there more solute in the cell, or is there more solute in the beaker outside the cell? So if this cell membrane right here, we're calling this a cell, it's just like a balloon, but cell membrane, we'll say it's only permeable to water. So none of these, the sucrose, glucose, fructose, those are staying where they are. They're either staying outside the cell, which is these three at this concentration, or they're staying inside the cell at this concentration. And if you don't know what M is, it's just a unit of concentration. So 0.03 molar sucrose just means there's 3 units, 0.03 units. Just think of it that way of solute. Okay, so does anyone have a guess as to where water will move? Will it move into or out of the cell? Into, yeah, exactly. So for those of you who are like, what? I don't understand. All you have to do is add up the concentrations. So you have 0.03 and 0.02. It doesn't matter that they're different solutes. It just matters that how much total solute there is inside versus outside the cell. So here we have 0.05 molar, 0.05 units of solute. That's all you have to worry about. Out here, we only have 0.03, right? So water wants to move towards where there's more solute, which is into the cell. So water is going to move into the cell. And then would you consider the environment, which is this beaker of solution around the cell, hypo or hypertonic? That might be the tougher question. So if we know the environment has less solute than the cell does, what does that make the environment? Hypo. I don't know if you guys said hypo. Okay, I just heard high. Hypotonic. So hypo means low. Remember that. So the environment is hypotonic. It has less solute in it than the cell has inside of it. So that would make the cell hypertonic. Like I said, it's always one or the other, and whatever the one is, the other is the opposite. They're always in relation to each other. So the environment is hypotonic, and the cell is hypertonic. So if you can do something like that, you pretty much have a good understanding of what this idea of osmosis and diffusion is dealing with. So I have some homework. I'll hand this out at the end of class for you guys in person. I posted it on Canvas, but there's some diffusion and osmosis questions to work through. They're pretty simple. It shouldn't take you that long. So it'll be due on Monday. And just it hopefully will help kind of solidify this sort of water movement between the cell and the environment, vice versa. All right, any questions about this? Okay. Now we're going to switch, not switch topics. We're still talking about stuff moving across cell membranes. But we're going to move into the active transport categories. So far, we have talked about movement across the membrane that doesn't require any energy. No energy required for any kind of diffusion, simple or facilitated. Active transport, this little lightning bolt of ATP here indicates that we need some energy to make these particles, these squares and circles, move across the membrane. So that's what we're going to talk about now. If you remember with the passive transport stuff, we're moving with the concentration gradient or down the concentration gradient, which means the particles that are moving, we're moving from an area where there's a lot of them, high concentration, to the other side of the membrane where there wasn't as many of them, to low concentration. So high to low concentration is down or with the concentration gradient. That's naturally how particles want to move. That's why you don't need any energy. They're just going to do it naturally. Active transport, as you might imagine, now we're going to go the opposite of what particles naturally want to do. So we are going to move particles against or up the concentration gradient. So they're going to move from areas of low concentration of themselves to areas of high concentration. And that's why we need energy, because they're not just going to do it naturally. They need a little bit of help. So it requires energy. We're just going to assume all energy is ATP. It's not necessarily the case, but ATP is the main source of energy. So it's going to require energy or ATP. Those will be interchangeable for us, energy and ATP, same thing. There's a lot of examples of this. Obviously, I don't need to give you every single example, I'm going to focus on one in particular that is really critical and actually takes up about 50% of the daily calories that you burn every day. So in your body, you're burning, I don't know how many calories you burn in a day. 50% of those are going just towards what we call the sodium potassium pump. So this is moving sodium particles, sodium ions, and potassium ions across cell membranes. That takes up half of the calories of your daily burning. So let's talk about what that is and kind of how that works and how energy is needed. Okay, has anyone heard of the sodium potassium pump before? Oh, okay, awesome. A couple people have. All right, so these are the basics. I'm just gonna go through the basics of what the sodium potassium pump is. We're talking about moving sodium, for our purposes, out of the cell and potassium into the cell. So keep that in mind. They're going in opposite directions. So outside the cell, when we start, we already have a lot of sodium. So the sodium concentration is high outside the cell. We're trying to kick even more sodium out, essentially. So we're moving more sodium to where there's already a ton of sodium. That is going to require ATP. So this is our little helper protein, the transport protein, the blue thing. Those will take on 3 molecules or 3 ions of sodium. So with each pump, so you can think of this as one pump cycle, we're going to kick out 3 sodium ions using ATP because we're moving against the concentration gradient. And then they'll be released and let out into the extracellular or outside the cell fluid. So sodium concentration outside the cell is high, inside is low, but we're moving sodium from that low to high concentration. 3 sodiums leave in each cycle. Okay, so you want to know those main points. I tried to boil this down to the main points of the sodium potassium pump. This is happening in most of your cells all the time. Like right now, you're pumping sodium out and you're pumping potassium in. Okay, so we've got sodium leaving the cell. That's where we stopped on the previous slide. Now let's deal with potassium, which is K+. Potassium is just the opposite. So outside the cell, there's not a whole lot of it. And inside the cell, it's in high concentration. But we want to bring more potassium into the cell, even more. So build up the concentration of potassium inside the cell. Only 2 potassiums come in with each sodium potassium pump cycle. We still need energy, so energy is necessary. That's what this is showing. Don't worry about that, but it just means energy is being used to bring the potassium in. It is the same protein, so you'll see here, over here we have the sodiums. On the left, that's where they bonded and then got kicked out. On the other side, the potassiums bond. This is kind of a simplification, but it's the same protein. That's the take-home message that's doing all of this. Okay, so the potassium comes in and gets released into the cell where there's a high concentration. So we're moving again up the concentration gradient, low to high. And we have 2 potassiums coming in each cycle. 3 sodiums go out, 2 potassiums come in. You want to have those numbers burned into your brain. Maybe not burned in, but memorized. So all in all, that's the sodium potassium pump. We're moving sodium out, we're bringing potassium in. We're trying to build up concentrations of each of those. More sodium outside the cell, more potassium inside the cell. So here is just an image of all of those plates put together. So the entire cycle. 3 sodiums are going to bond with the protein. We'll use some ATP to make the protein basically change shape. So it pushes the sodium ions out. It's still open, so potassium ions come in, use a little more energy, and then the potassium ions are released into the cell. 3 sodiums for every 2 potassium. So I put that down here too. All right, so what? Why do we care so much about the sodium potassium pump? Well, you may not, but biologists in general care a lot about the sodium potassium pump, and I guess you should. Like I said, it's burning half your calories, so why is it so important? Okay, so let's think about the charge difference here. So both of these are positive ions, right? So sodium is positive, potassium is positive. If we're kicking 3 positive things out and only bringing 2 positive things into the cell, what does the charge become inside the cell? Positive, negative, or neutral over time? Negative, yeah. So if you're kicking out more positives than you're bringing in, that's going to bring the charge in the cell down, right? Because these are positively charged molecules. So if you have fewer and fewer positively charged molecules in the cell, it's becoming negative. So inside all of your cells, for the most part, when they're resting and not doing a whole lot, they're actually negatively charged inside. That's called the resting membrane potential, or electrochemical gradient. So there is this charge differential. So we have a negative charge inside our cells, thus it's kind of positive outside our cells. So why would this matter? Does anyone have any idea? Any idea what it relates to? It's okay if you don't. Okay. So that basically creates this voltage, which is crazy to think about in your cells, but it creates this voltage differential inside versus outside your cell and is building up energy. So that negative charge inside your cell, as it gets more and more negative, it stops at a certain point once it gets so negative. But that's essentially, you can think of it as a ton of potential energy. And when you want to contract a muscle or one of your neurons, like your nerve cells fires, that potential energy is essentially a release. So you have this opposite flow of ions and electricity is basically running through your body because you have this charge differential. It's just like a battery where you have a negative and a positive side. So this would be the negative side inside the cell. This would be the positive side outside the cell. When you release these ions, when you consciously think, I want to flex my arm, that's what's happening. You're sending an electrical current through your body because you've built up this voltage in your cells. It gets, obviously, that's a simplification, but in the end, it's all just electrical signals that are moving all of your muscles all the time, whether that's conscious muscles, like your skeletal muscles, or your heart muscles, your digestive tract muscles, all that kind of stuff. So we have to have this negative charge inside our cells, which is why the sodium potassium pump is so important. If we didn't have this charge differential, there wouldn't really be any potential energy to release. So hopefully now you think, oh yeah, I can see why the sodium potassium pump is important. It all comes down to that voltage that's created across the membrane. Okay, any questions about that stuff? We're kind of moving on to a different kind of transport that I didn't really talk about yet, but it's sort of always just talked about on its own, bulk transport. It's not, we're not involving like proteins specifically. It's a little bit, I think it's a little bit simpler to understand. So we good, no questions? Okay. So this last type of transport across the membrane is called bulk transport. There's a few different types that I'll talk about over these next few slides. There's two main types. You have exocytosis and endocytosis. Exo means out, endo means in. So exocytosis is basically moving molecules out of the cell, but a lot of them. So the sodium potassium pump will move, a few molecules at a time. Exo and endocytosis is bringing in a ton of molecules or kicking out relatively a ton of molecules. So that's why it's bulk transport. It's a lot of stuff moving into or out of the cell at one time. So exocytosis will essentially take these molecules that it wants to get outside the cell fairly quickly into these little vesicles or like little transport spheres. Those transport spheres will essentially bind with that plasma membrane. So you can see here, they all have this plasma membrane as well. And then inside there is the molecules they want to get rid of. So those two plasma membranes will combine and mesh and then end up releasing the molecules that are inside that vesicle. So I think you guys on Zoom could see that okay, but this is a vesicle with the basically phospholipid bilayer, same thing as the cell membrane. So this is going to be the cell membrane, sorry if that wasn't clear, that long thing. That is the cell membrane. And it will bind and bond and basically become more cell membrane and release these particles out of the cell. Endocytosis, just the opposite. So this plasma membrane will kind of cave in here. Those molecules that are within that caved in area will then end up in their own vesicle. So the plasma membrane will pinch off and become its own vesicle, bringing in kind of engulfing a lot of those particles that it wants to bring in all at one time. So here we have the plasma membrane bending inward, and then it'll pinch off right here and become its own vesicle. And then these will just go wherever they need to go in the cell. So that's exocytosis and endocytosis. That is all I'm going to say about exocytosis. Bringing stuff into the cell, there's actually 3 different types of that. So I'm going to go over those really quickly. They're pretty easy to understand. So remember, this is endocytosis. We're bringing stuff into the cell. Phagocytosis is probably the most well-known one, I guess. But that's where you have a big solid particle or multiple big solid particles that you want to bring in. So this particle would not be able to move across the membrane in a protein. It's just too big to actually have a protein able to bring it in. So that's why this endocytosis, specifically phagocytosis, is bringing it in. So a large solid particle, that's brought in by phagocytosis. Any kind of smaller molecules or fluids are brought in by what we call penocytosis. And that means cell drinking. So it's like drinking a bunch of the extracellular fluid, bringing it in along with whatever molecules happen to be in there. So phagocytosis and penocytosis, the difference is really just the size of the molecule. And then there's a very specific way of bringing different molecules in called receptor-mediated endocytosis. That just means there's these little receptors on the cell membrane that are only going to allow certain molecules to attach to them, which will then be brought in. So they're sort of keeping out these blue squares and only letting those stars in. So the squares are not allowed in, only the stars are going to bind to these usually proteins. They're usually proteins on the outside of the cell. Only the star can bind, so that means the cell can only bring those stars in. That's all it needs. So that's receptor mediated. These are the receptors, these little green horseshoe-shaped things. But it's still endocytosis because molecules are being brought in. So then all of these end up in some kind of vesicle. So then they'll move to wherever in the cell they need to go. Okay, what else do I have? That is it. And rather than jumping into the next chapter, I figure it's Friday, so we don't need to go into the next one. So we're going to stop there. Here's just a kind of review, make sure you understand this material. And on Monday, I have a whole day set aside for review for the next exam. So I don't think we'll be behind. I think the next exam will still be March 1st. And we'll talk about the cell cycle and mitosis Monday and Friday of next week. So there's no class on Wednesday, just Monday and Friday. And then we'll review for the exam on Friday as well. So yeah, that's it. You guys can go. We're done. No, it's okay.